if we are going to start about the antibody structure and its function. The antibody having that structure that given the diagram that I have to elaborate. First, the Fab fragment consisting of light and a heavy chain. They having the Fab fragment and that is consisting of light and heavy chain that recognizes the antigen. The Fc region of IgM and IgG fixes complement and heavy chain contribute to Fc and Fab regions. The light chain contributes only to Fab regions. And what are the Fab? The Fab are fragments antigen binding. To determine the isotype, unique antigen binding pockets, only one antigenic specificity expressed per B set. So they having the Fc, they will C's, 5C, they are the constant. This region, this is the Fc, okay, and that is the Fab region. In that, they are constant, they are the hydroxy terminals, they are complement binding, and they are having the carbohydrate side chains, and they are having a comfort that determines the isotypes, what they are going to, there will be IgM, IgD, etc. And if you guys make a concentration there, they are of VL and VH, light and the heavy chain with the constant for heavy chain and the light chains. And if you guys notice, they are having a one bond that is disulfide bond. That is very important that having the disulfide bond. And these Fc regions are complement binding sites and they are macrophaging binding site, this one. Now we are going to our definition of diversity. How they will be having a huge variable and diversity in the antibody generation. The antigen independent. They are having the random recombination of VJ, digen and VDJ habitation. You guys already know they have a domain, constant and different thing. Their genes are regulated by RGA1 and RGA2. They're having the random addition of nucleotides to DNA during recombination by terminal deoxynucleotide transferates and random combination of heavy chain with light chains. They are having a generation of antibody specificity, the antigen dependent. They are having two type of generations. One is independent, that having diversity, and second generation of antibody specificity. The diversity is independent from the antigen and specificity is dependent on antigen. The antibody directly combine with the antigen that they needed. They are specific for the antigens and they are somatic hypermutation and affinity of maturation of variable agents. They have an isotype switching with a constant agent. You guys take that they are the neutralizations. The antibody prevents the bacterial adherence. Opsonization, antibody promotes phagocytosis by marking pathogen for destruction. They activate the complement system by the membrane attack complete MAG. And antibodies activate the complement enhancing opsonization and lysis. About the immunoglobulin, they are having different isotypes. By the simple formula, GAM, you can remember as IgG, A, M, D, and E. To start the elaboration, the all isotype can't exist as monomers. It is wrong. The all isotype can exist as monomers. They can exist separately in a one molecule white structure. And there is a matured native B cells pure to activation express that IgM and IgD on their surfaces. The mature and a named B cell pure to activation express to IgM and IgD on their surfaces and they may differentiate to germinal centers, often known by isotype switching and G rearrangement that induced by cytokines and CD40 ligand. The switching are totally the gene rearrangement and that is going to induce by cytokines and CD40 and into plasma cells. They secrete IgA, G and A. For B cells, IgM and IgD mature to plasma cell at day H. They are saying for the B cell, IgMOM and that full mom and for that mature to plasma cells and they go for the H's. And what are the H's stand for? IgA, G and A. It's a very good mnemonic. The moment that IgM and D are giving and maturing the B cells and that will converting into plasma cells and these plasma cells are going to secrete IgA, G and the E. Affinity refers to individual antibody-antigen interactions, while avidity describes the cumulative binding strength of all antibody-antigen interactions in a multivalent molecules. First, we have to talk about the IgG. The IgG is a main antibody in a secondary response to an antigen. It's a more abundant isotype in serums, and it fixes complement opsonization of bacteria, neutralizes bacterial toxins and viruses. Only isotype that crosses the placenta provides infant with passive immunity that start to wane, but it's very important. It's the only one and only exception that pass the placenta. IgG greets the growing fetus. Like IgG is going and say hi to the fetus, like you are in our body. And associated with the warm autoimmune hemolytic anemia, the warm weather is G. When they will be very increase in temperature like that, they will can cause the hemolytic anemia. And therefore, again, the warm weather is good. And then the IgA. The IgA prevent the attachment of bacteria and virus to the mucous membrane to prevent the destruction of a normal structure and does not fix the complement. The complement they are going to fix is by IgG. And there's a monomer in circulation or dimer. They can do monomer and can be dimers with the chain which when secreted and crosses epithelial cells by transcytosis. They can cross the epithelial cells by transcytosis and produce in GI tract by pyrobatches and protects against gut infections. And most produce antibody overall with IgA but has a lower CM concentration. Maximum that antibody are going to produce is IgA, but is having low CM concentration and release into secretion like tears, saliva, mucus, and plasma, and picks up security component from epithelial cells which protects the FC region from luminal proteases. And guys, this next is IgM, the mastermind. The first antibody to produce during an immune response is that fixes the complement antigen receptor on the surfaces of the B cell. The monomer on B cell, pentamer with the J chain when secreted. They are going to be normalized in the pentamers, but they can be monomer on B cell and it is secreted with the gene. Pentamer enables avid binding to antigen while hormonal response involved. It associated with the cold autoimmune hemolytic anemia. The cold autoimmune hemolytic anemia IgM and warm autoimmune hemolytic anemia is associated with IgG. The warm weather is good. Cold weather is mastermind. It's IgM. The IgG is expressed on surface of mature named with the normal low level are detected in serum. IgE binds mus and basophils, crosslinks when exposed to a virgin and mediating entity through release of inflammatory mediators such as histamine and contribute to immunity to pass activating the eosinophils. And also it is called the antibody-dependent cytotoxicity like activation of any cell by using an antibody that is eosinophil using IgE. Same like IgG is using by natural killer cells sometime. This type NMM, the thymus independent, thymus dependent. What is happening? The antigen liking a peptide component, while lipopolysaccharide from gram-negative and can't be presented by MSC2 T cell. And with the immunogenic vaccine often require booster and adrenaline, that is the capsular polysaccharide subunits of Streptococcus pneumonia. And sometimes the antigen containing a protein components and that cause the class switching and immunological memory occur as a result of direct contact of B cell and T helper cells. That is thymus dependent and thymus independent having the liking peptide components that can go to present by MSC2 class T cells. Fine.